And as we are studying about genetics, so our first important term is gene. So gene is the forms of inheritance. That means it is the functional unit of inheritance. It is functional unit of inheritance. And the term gene is given by Johansson. It is given by Johansson and it is commonly known as factor, and the term factor is given by Mendel. It is the basic of your gene. Nextly, allele. Allele is the forms of gene. It is the forms of gene. And most commonly, there is two forms of gene. One is your dominant and another one is your recessive. So, your first one is your dominant and second one is your recessive. If we take two examples of a plant and its height, here it is your tallness, then the dominant letter which is represent it is represented by capital T, and in same case, if we take an example of dwarf, then the representing letter is your small t. So here we look that in dominant the tallness is your capital T, but in recessive it is dwarf, that means short, but the representing letter is not S or D, the representing letter is your small t. So here the forms of the gene, that means your capital T is a form of gene and small t also a form of gene. That means capital T represents the tallness of the plant and small t represents the dwarfness of the plant. That means these are the alleles which represent the character. That means your tallness and the dwarfness. And we also take another example combining both two as your capital T and small t, and couple of stages later we just discussed about these things. Nextly, the third important term is your homozygous condition. In homozygous genes or homozygous condition, we found two genes of same type. That means if we take dominant, both the genes are dominant, that means both are capital T. If we take recessive, then both are recessive, that means both are small t. So it is your dominant trait, it is your recessive trait. Homozygous means both are same. Homo, here is both are same, here also the both are same. So it is your homozygous condition. Heterozygous condition, next important term. In heterozygous condition, we found combining two terms. That means one from your dominant, one from your recessive. That means we just combine over here. It is your capital T, and the one is your small t. It is your recessive trait. That means it is your heterozygous condition. That means your capital T and small t. But homozygous condition, capital T in dominant, and capital T, small t in recessive. It is the basic difference of your homozygous and heterozygous condition. Nextly, the phenotypic and genotypic recessive. So our next important term is phenotypic and condition means physical appearance. Here we talk about the physical appearance of a trait. Nextly, in genotypic condition, we just discuss about the genetic makeup. We just discuss about the genetic makeup. Let's take an example first. So, if we take an example of your capital T and small t, that means it is your tall plant. Physically, if we look this plant, we say that it is your tall plant. Nextly, another example. Capital T with capital T. So, both are dominant character. That means it is your homozygous condition, it is your heterozygous condition. We just wrote over here. So, here also it is your tau. In both the cases, if we judge about the physical appearance, then in both the cases the plant is tall. But if we talk about the genetic makeup, that means the genetic appearance, then we talk it as, that means then we talk the capital T small t as heterozygous condition. That means it is your heterozygous tau. So the plant is your heterozygous tau, and we wrote it as capital T and small t over here. So it is your heterozygous tau. Nextly, it is your homozygous tau. So we wrote it as capital T and capital T. So it is the genetic change. That means we genetically represent it as your capital T small t and capital T. But in physical it is both are tau. So it is otherwise known as your phenotypic and it is known as your genotypic. It is the basic difference. Nextly, we talk about the cross. So our next important point is cross. Cross means basically in mathematics this is your cross. That means it is just the symbol of your cross. But in biology, cross means we just cross to produce a new thing. That means if we take an example of a tall plant, reproduce with a short plant, and we form a heterozygous condition or a homozygous condition. So these types of things, when we cross between two different characters, then the alleles are always known as a cross. And for the crossing, we just use a number of terms, and couple of stages later we just discuss about that. So it is basically known as a cross. Cross means just mixing of two, that means breeding of two characters, basically known as a cross. So our next important term is a filial. Filial means in monohybrid condition, dihybrid condition, trihybrid condition, again we discuss about your crossing. Then in these conditions we just found some of the alleles after crossing, and the alleles are first alleles which are found for the first cross is known as the F1. Later on we just discuss about what is your F1, what is your F2. So we just wrote is an F1 and F2, F3, so this F1, F2, F3 is the resultant of a generation. So it is otherwise known as filial of first generation. Nextly, it is again crossed and forms your filial of second generation. Then again crossing is takes place. Then the resultant is forms your filial of your third generation. So these are the resultant products which are otherwise known as your filial. And the next important term is your Punnett. Punnett square is just a checker board. So it is just a checker board. You just erase it. It is just a checkerboard type where we cross two type of characters. That means in both the dominant character or both the recessive character, we just cross these things and get some of the resultant or some of the products. So the next type is your Punnett square. Punnett square is just a checkerboard. We just take over one example of capital T is processed with small t. It is just a checkerboard. So first we draw a checkerboard over here. So it is your male set. Suppose it is your female. Just wrote the gametes first. It is one gene, and these are the ladies. And from this, these are the alleles. If we plot it in a Punnett square, it is your capital T from the male set and small t from the female set. Next thing we just cross it for capital T and small t. Here we get the capital T, small t, and in every set we just get the same answer. The capital T and small t, capital T and the small t. So we just plot a Punnett square, and after crossing we get the resultant, and where we get the resultant of same particular type of genes, we just cross it and get the resultant over here, and the graph is already known as a Punnett square. That means the checkerboard type thing is already known as a Punnett square. Next thing we just discuss about the things, that means the condition of your selfing. So our next important term is your selfing. Selfing means if we cross both the genes of your same, if we take an example of this checkerboard and cross the capital T with that capital T with the capital T, then the crossing is always known as your self. Self means in the same type we just cross two things. That means for the capital T is crossed with again the capital T of the same generation. Then it is known as your selfing. Selfing means self cross. Nextly, the important term is your purity. So what is your purity? Purity or your pure line? Pure line means the purity condition. That means if we take one example of capital T is crossed with another capital T, and the resultant is mostly the capital T and capital T. Here we just cross a same trait again and again. If we repeat from the filial 1 to filial number 5, from first generation to fifth generation, if we get the same resultant in every generation, then this trait is always known as a pure line. There is no other things are there. That means this seed is your pure line seed. That means the plant is produced over the seed is the pure line plant, is always known as your condition. That means this is your pure line condition, where we get the same offspring from the first generation to the fifth generation, up to the fifth generation commonly, then it is known as your pure line condition. We get the same answer in every condition. So it is your pure line. Nextly, the cross. In cross, we get your monohybrid cross, your dihybrid cross, and the trihybrid cross. We just shortly discuss about what is your monohybrid cross, what is your dihybrid cross, what is your trihybrid cross, and again couple of classes later we just discuss briefly what is your monohybrid and what is your dihybrid and some of the laws of your dihybrid or monohybrid cross. That means Mendel gives a number of laws also. So just discuss about the point number 11, monohybrid cross. Monohybrid means, mono means your one, Cross means your cross. That means we cross a single character over here. That means capital T, capital T. Just consider it as it is the height. It is the tallness and it is just the height. We just take consideration with a single character. Monohybrid means we just cross with a single character. Nextly, that means again also your small t. So it is your short. That means here we cross a single character of capital T with a single character of your small t with small t. So it is about your monohybrid cross. Monohybrid means single character just crossed. Nextly, the dihybrid. Dihybrid condition means cross means we just get two characters. Consider that di means your two, we just get two characters. One is your capital T, and another character is your color. That means your red color. Consider here capital R. That means it is your height, which is tall. It is your color, which is red, and it is crossed with the small t with smaller. That means it is also the height, but the height is short here. It is also the color, but the color is white here. We just take an example. So here we cross two type of characters with two type of character. Here we cross one character with one character. If we cross one character with one character, then it is your monohybrid cross. If we cross two character with two character, then it is your dihybrid cross. Next your trihybrid cross. Same as it is. Trihybrid cross means trihybrid cross means your three characters. If you take an example of your capital T, cross with your capital R, sorry, it is the single trait, capital R, and another is your seed cell. That means you may also take an example of the appearance of the flower. Examples you may take. That means it is your height, it is your color, we already taken over here, and it is the axial position of the flower. In the other hand, if we cross it with the small t, with the smaller and smaller. That means here is one character height, another one is your color, another one is your axial. That means the position of the flower. And again we cross it the short and the polar and respectively the position of the flower. So whenever we cross three type of things in a single trait, then it is known as your trihybrid cross. When we cross two type of things, then it is known as your dihybrid cross. If we cross a single trait with a single, then it is known as your monohybrid cross. So it is all about your cross, and there is a number of other important terms are also there, but we just discussed some of the basic important things. But so hope this session is helpful to all of you. Till then, stay tuned. Thank you.